Okay, so I want to just review what you need to know about experimental design and setting up an experiment. One of the things that you're going to be asked to do on your assessments is to evaluate a scientific setup or an experiment. So what you need to look for is you need to look for your control group and your test group. And I'm just going to use the example that we used in class with the mussels, okay? So when the students set up their experiment, they set up an experiment with 2 tanks, both tanks being the same size. They put the same amount of water in each tank. They put the same number of mussels in each tank. They made sure that the water was the same temperature. They made sure that the mussels were of the same type. And all of these things are called variables, okay? So variables are the things that we use to evaluate a testable question. So in this case, the tank, the water, the mussels, those are all variables. Now, when we keep all of the variables fair or the same, we make sure that everything is exactly the same. That is called controlling our variables. And you control variables so that you have a valid experiment. If you don't control your variables, then you probably won't be able to test your hypothesis. You probably won't be able to test it and find out what is causing something. So we need to make sure that all of the variables are controlled. We do, however, need to change one thing, and the one thing that we need to change is what we're testing. So for example, in this experiment, we were testing to see if phosphates were causing the mussel population to decline. So that one variable, the one that we change, is called our independent variable, okay? And the independent variable is the one thing that we change. Now what I'd like you to do is have a mental model of that. Do you remember when I held up my finger? One thing we change is the independent variable, okay? So in this case, the independent variable is adding phosphate. So the phosphate is the independent variable. That's the one thing that we change. Now, we also talked about test group and control group. Here's what you need to remember. The test group is the one that has the independent variable. So this one is the test group or the experimental group because that's the one that we're testing. We are testing the phosphate to see if it causes a decline in the mussel population. So this is called our test group. Or our experimental group. You can use either term interchangeably. So the test or experimental group. And you need to be able to distinguish these. Okay? The other container, the one that we didn't change, the one that we kept everything the same as in here except for that one thing, this one is called our control group. And the control group is typically the one that is closest to the natural conditions. And what we tried to do was mimic the natural conditions of the mussels by making the water the temperature that they are normally exposed to. We didn't change anything about what their normal experience would be. And the reason that we need that control group is because we need to be able to use that to compare the experimental group against. So we look at this group and compare it to this group. Now, the last thing that we need to do is we need to measure the results. And the way we do that is we collect our data, okay? And our data, in this case, would be the number of mussels that died in each group. Now, you guys remember when we did the experiment, or we pretended to do the experiment, that in this group, some mussels did die. And it makes sense because they're living organisms, and living organisms do die. But we also had to look at whether or not the same number of organisms died in this group. And what we found that our hypothetical data was that about 600 out of about 10,000 mussels died in this group. But we found about 1,800 out of 10,000 mussels died in this group. And in order to collect proper data, we have to remember that, so naturally, about 600 mussels will die. So we have to take that number out of this number so that we get an accurate picture of about how many mussels did not die of natural causes. So about 1,200 mussels did not die of natural causes. And we might be able to say that, well, it could be the phosphate that is causing those other 1,200 mussels to die. And the number of mussels that die, that's our data. And we call that data, or the amount of the information that we collect, we call that the dependent variable. Okay, so those are all of the parts of the experiment that you need to understand.